This is the Kreb cycle, the second step in cellular respiration. For every glucose molecule, the Kreb cycle runs twice because glucose is a 6-carbon molecule, and each turn of the Kreb cycle breaks down 3 carbon molecules.
So the inputs and outputs of the Kreb cycle. Inputs are 1 ADP, 1 FADH, and 3 NAD+. The outputs of the Kreb cycle include 3 NADPH, 1 ATP, 1 FAD, and 2 carbon dioxide molecules. So step 1. Glycolysis produces a 3-carbon pyruvate molecules, which are then broken down by an enzyme and converted into acetyl-CoA, a 2-carbon molecule, releasing carbon dioxide. The acetyl-CoA is then bound to oxaloacetate, a 4-carbon molecule, by the process of citrate synthase to produce citrate, a 6-carbon molecule, releasing water. This process helps bring in enough carbon atoms to initiate the Kreb cycle.
So when citrate loses one water molecule, it converts it into isocitrate, which is another 6-carbon molecule. And isocitrate is then oxidized, which produces the 5-carbon molecule alpha-ketoglutarate. And this reaction releases electrons, which end up reducing NAD plus into NADH and releases CO2 while that happens.
The enzyme alpha-ketoglutarate dehydrogenase then converts alpha-ketoglutarate to succinyl-CoA, a 4-carbon molecule. An input of NAD plus is required for this step, and the process produces 1 molecule of NADH and CO2, as well as a hydrogen ion. This CO2 molecule is where the 5th carbon goes, making succinyl-CoA a 4-carbon molecule. The succinyl-CoA just created is then converted into a succinate, a 4-carbon molecule, by an enzyme called succinyl-CoA synthase. This process yields 1 ATP molecule.
Succinate of 4-carbon molecule becomes fumarate of 4-carbon molecule through action of the enzyme succinate dehydrogenase, and FAD is reduced to FADH2. Succinate dehydrogenase is a direct part of the electron transport chain. It is also known as electron carrier 2. Fumarate is then converted to malate, another 4-carbon molecule, by incorporating water with its use of fumarase. Malate is converted to oxaloacetate, a 4-carbon molecule, by malate dehydrogenase, and NAD plus is reduced to NADPH. And with the oxaloacetate, we're back where we started, ready for acetyl-CoA to be bound to it.